This is Module A5, Objective 3. We're going to look at the various types of neuroglia cells, including their locations and functions. Neuroglia are cells that support the neurons. There are, if you look at the number of neuroglia cells compared to that of the neurons, we see that half the volume of the nervous system is composed of the neuroglia cells, and they far outnumber neurons as a whole. There are many types of neuroglia, both found in the central nervous system as well as the peripheral nervous system. The peripheral nervous system has two types of neuroglia. We have the satellite cells and Schwann cells, while the central nervous system has four types. We have ependymal cells, microglia, the astrocytes, as well as the oligodendrocytes. Ependymal cells are one of the cell types found within the central nervous system, and these cells, they line the central canal of the spinal cord, as well as the ventricular system of the brain. And if you have not had these anatomical structures in the lab, you will soon. The ependymal cells secrete the cerebrospinal fluid, the CSF. They're often ciliated to help circulate that cerebral spinal fluid. Remember, the function of cilia is to move a fluid over the surface of the cell. So as the surface of the cell, all these cilia, as they beat in one direction, they're going to actually move the fluid over the surface of the cell. Ependymal cells monitor the cerebral spinal fluid, and they do contain stem cells for repair. Astrocytes, they help maintain the blood-brain barrier, which isolates the central nervous system from the bloodstream. This is relatively important because various compounds dissolved in blood need to be separated from the nervous system. If you consider the hormones, the amino acids, and the other chemicals that fluctuate in blood, these chemicals could ultimately affect neuron function. So it's very important to keep the interstitial fluid of the central nervous system separate from the bloodstream. And astrocytes do that. They have little extensions off of their cell bodies that expand into little foot-like pads that literally coat and cover the blood vessels, preventing free access, preventing diffusion from circulation to the interstitial fluid within the central nervous system. Astrocytes have a tremendous amount of filaments that help create the three-dimensional framework of the central nervous system. They can repair damaged neural tissue to some degree. During development, they have a very important role of guiding neuron growth and development. They help guide neurons as they make attachments to other neurons. Down here we have a histograph of astrocytes. You can see they're very numerous, and again, they're going to create this three-dimensional framework, so they need to be. The oligodendrocytes, these cells have processes that extend from the cell body. The very long, flat, kind of pancake-like structures that will wrap themselves around an axon, creating what is known as the myelin sheath. Myelination, and here in this electron micrograph, we see all the layers surrounding this axon. Again, this is all the extension of that oligodendrocyte, and this layering effect insulates the axon, and this insulation actually increases the speed of the action potential. We'll talk in the next objective about why this is important, for now, just memorize that the myelination due to the oligodendrocytes in the central nervous system increases the speed of the nerve impulse or action potential. That myelination is largely lipid. About 80% of myelin is composed of lipids, and this gives a white glossy appearance under dissection. White matter, and I'm sure you've all heard of white and gray matter of the brain. The white matter is areas of the brain that are largely myelinated. That glossy white coloration from the lipid content of the myelin gives those regions of the brain the white glossy appearance, and it's referred to as white matter. Gray matter, on the other hand, is not myelinated, or there's not a lot of myelination, and the neural tissue just looks gray. Demyelination, the destruction of myelin, is associated with a number of pathologies. You have a clinical note. Let me see if I can get the page. You have a clinical note on page 397 that covers various pathologies associated with demyelination. Down here in this picture, we see that you have a normal nerve, and underneath we have a nerve that is affected by multiple sclerosis, and we can see that the myelin sheath is being damaged. What would this do? Well, again, we need to consider the function of myelination, and we had just stated that myelination speeds up an action potential. So a damaged myelin sheath would slow the action potential down, and this causes the major symptoms of multiple sclerosis, heavy metal poisoning, and Guillain-Barré syndrome. With myelination, you end up creating a system of nodes and internodes down the length of the axon. The internodes would be these regions where myelination has occurred. So all of these regions of the axons are internodes. And what that leaves is an area that is the node. And the nodes exist between the internodes. Let me get a good color. So here we have a node, and this is also known as the node of Ranvier. And these nodes are regions of the axon that are unmyelinated. And again, this is going to come in and be a very important component of the material that we cover next. The relationship between the nodes and the internodes. The last neuroglia of the central nervous system are the microglia. And these cells are roaming phagocytes. They simply migrate through the central nervous system, phagocytizing any type of waste product, debris, pathogen that may be present. So these are active immune defense cells of the central nervous system. They're often described as being the garbage men because they simply roam around looking for any type of garbage, if you will, and they phagocytize that material, break it down with the lysosomes and the other types of digestive enzymes found within the cell, and they dispose of the threat. As we move into the peripheral nervous system, we have two types of neuroglia. We have the satellite cells and the Schwann cells. Satellite cells are associated with the ganglia. And if you remember, within the peripheral nervous system, I talked about ganglia, these little expanded pouches along a nerve. And this is where the cell body of the neurons are found. So the cell bodies of many neurons are found. And if you think about the shape of a neuron, you have a very large cell body, and then you have the axon, which is this little very thin, long extension. So when we consider a peripheral nerve, the cell bodies tend to hang out in the ganglia, and their axons simply run the rest of the nerve. So satellite cells are closely associated with these ganglionic regions, and they're going to help regulate the environment around the neuron, around that cell body. Schwann cells is the last neuroglia that we're going to look at, and again, these are found within the PNS, and they form the myelin sheathing around the peripheral axons. Oligodendrocytes can actually send extensions, and they can myelinate many axons at a time. One of the main differences between the oligodendrocytes and the Schwann cells is that Schwann cells can only sheath one segment of an axon. So they don't have the ability to sheath multiple axons. They really do dedicate themselves to one. These are two different situations. Here we actually have a Schwann cell starting the process of myelination. They wrap themselves around an axon, and as we progress, you see that myelination, or that sheath, becomes increasingly thick, and it's going to better insulate that axon. So you have one Schwann cell associated with one segment of one axon. We do have a case, however, where Schwann cells can wrap themselves around a number of axons. They're not myelinating. You don't have the multiple layering effect, but they do help bind and support a number of axons. But again, it does not deal with myelination. When you think of Schwann cell, you should think of myelination in the peripheral nervous system, and that one Schwann cell is associated with one segment of one axon. This is a kind of another service that they perform, but don't get caught up in that, because I don't want that to confuse you. This is a figure out of your text, and again, it just kind of shows the close association between the actual neurons and the neuroglia. Up here we have the ependymal cells. What is the function of the ependymal? Again, these cells create the cerebral spinal fluid. They monitor the cerebral spinal fluid, and they help circulate with the cilia associated with their, let's just draw some cilia on these cells. And as these cilia beat, they're going to help circulate that cerebral spinal fluid. Here we have an astrocyte. Again, astrocyte, because these cells are kind of stellate, they look like stars. Here we have the extensions of the astrocyte literally closing off a blood vessel, creating the blood-brain barrier, the BBB. And they're also closely associated with neurons, as they help support and guide the growth early in development. This little critter is a microglia. And the microglia are the garbage men of the CNS. They're the active immune cell that circulate and try to phagocytize debris. We have the oligodendrocyte. These are the myelinators of the central nervous system. And I know this is central because one cell is wrapping itself around two different axons. Here's another oligodendrocyte. What is the function of this myelin coating? It increases the speed of the action potential, or nerve impulse. Here's another oligodendrocyte. This particular diagram is separating white matter. So this lower figure is white matter from the gray matter up above. So you can see that there's a lot of unmyelinated structures in the upper portion of the figure, and that would refer to the gray matter. I don't think, looking over this, that I've left much out. I mean, we can identify the inner nodes. Here's an inner node versus the node. That will be discussed later. But go ahead and look. This is on page 394 of your text, and you can kind of look through that and find those cells on your own. So if we were to sum up neuroglia, we see that they perform all the communication, the processing. Oh, I'm sorry, I said neuroglia, neurons rather. They perform all the communication, the information processing, and they basically control the functions of the nervous system. Neuroglia, they preserve both the biochemical and the physical structure of the neurologic tissue, and basically they're there to help support the neurons.